Buenas tardes, ya son tardes. Bienvenidos. Esta es la segunda conferencia de la 12ª edición de este panorama. Y hoy tenemos la presencia del doctor Wexler, Anthony Wexler de UC Davis. Quisiera decir algunas palabras antes. La organización que vamos a tener para esta conferencia es que el doctor Wexler va a darnos su conferencia. Vamos a dejar las preguntas o aclaraciones hasta después de que él termine, para entonces tener un periodo de aproximadamente 15 minutos de preguntas. Y entonces, por favor, todos los comentarios que tengan, guárdelos para cuando él termine. Como dije, esta es la decimosegunda edición de este panorama. Esto inició hace 12 años. La directora Amparo Martínez fue la que inició este panorama. Lo continuó la doctora Telma Castro, a pesar del COVID. Y está ahorita siendo apoyada por Jorge Zavala, con el esfuerzo de muchas otras personas. Y yo creo que es uno de los foros científicos con un gran contenido científico que tenemos ahorita en la universidad. Así que trasciende nuestro instituto, y yo creo que es actualmente una de las conferencias de más alto nivel y contenido científico que tenemos en la universidad. Conocí a Anthony Wexler creo que por casualidad, en una conferencia que después me enteré que él tiene mucho que ver en su organización en Riverside, en Pasadena, hace 4 años. Y en donde de hecho la persona que se acercó a mí fue el doctor Wexler, porque vio en mi gafete la palabra México. Y entonces por eso se acercó él a platicar conmigo, y desde entonces lo conozco. Él y su esposa llegaron desde la semana pasada a México. Su esposa está aprendiendo español. Y entonces tenemos una persona que no solamente aprecia nuestra cultura y nuestra ciudad, sino que también la goza muchísimo. ¿Okay? Entonces me da mucho gusto que Anthony Wexler esté ahora con nosotros. Anthony Wexler es el director del Centro de Investigación de Calidad del Aire y profesor distinguido de Ingeniería Mecánica y Aeroespacial, Ingeniería Civil y Ambiental y Recursos de Tierra y Aire y Agua en la Universidad de California en Davis. Su investigación se centra en procesamiento atmosférico y los efectos en la salud de la contaminación del aire. Obtuvo su título de bachillerato en ciencia en la Universidad de California en Berkeley, su grado de maestría en ciencia en el Instituto Tecnológico de Massachusetts y su doctorado en el Instituto Tecnológico de California en Caltech. Su investigación actual incluye los efectos en la salud de la contaminación del aire relacionada con el tráfico, los efectos en la salud de los sistemas de suministro de nicotina, la termodinámica de soluciones acuosas altamente concentradas, instrumentos para medir toxinas en el aire y compuestos biométricos para la captura de carbono. Y de hecho, está trabajando con nosotros en un proyecto PAPIT sobre instrumentación para medir contaminación del aire, en específico metales pesados. Entonces, le doy la palabra a Anthony Wexler, para que nos dé su conferencia. It's all yours.
Thank you. And thank you for wearing the tie with the colors of UC Davis. So welcome. Thank you for inviting me here. A wonderful crowd. Is there any seat available? There's a couple of seats available if you want to sit. If you put your hand up, if you're next to a seat that's available. There's only one over there, I think. You don't want to sit down? You're okay? Okay. Okay. So before I start talking about the neurological health effects of air pollution, I want to thank the colleagues that help me do all this work, and there are many of them. I am not a neuroscientist, and so I rely on neuroscientists to help me with that part of the work. So the first one here is Pam Line, and she is a neurotoxicologist, works on many different areas of neurotoxicology, and of course we're longtime collaborators on the work I'll talk about today. She mostly works on, Pam mostly works on the pathology, what's going on with the tissue. Jill Silverman, also at UC Davis in the School of Medicine, she works on the behavior. And so for some of the work we'll talk about the behavioral endpoints and the neurological health effects. Keith Vine, he has been with me. He started his PhD with me when I arrived at UC Davis in 2000, and he's been with me ever since. And Chris, and he designed the exposure facility, which I'll talk about, and he and Chris and I and other people built it. Chris Wallace is my lab manager and has been with me for more than 15 years. I don't even know how long it's been. Barbara Maher recently joined us. She's retired from the University of Lancaster. I'll be showing you some results from her work, which show the chemical composition and location of the particles in the tissue of the brain. Incredible. She's published in this area a lot already. And when she saw our work, she reached out to us to collaborate. And then many students and postdocs have worked on this and continue to work on this effort. And of course, funding. So National Institute for Environmental Health Science, National Institute of Aging, National Institute of Occupational Safety and Health, Mental Health, Child Health Development, whatever. All this funding makes all this work very expensive work that we're doing. Okay. So Steve did a wonderful talk yesterday, and I have a large act to follow, a difficult act to follow, because he did such a great job with the talk yesterday. And he gave an introduction to the Gerg Center at Texas A&M, and I'll give just a one-page slide here introducing the Air Quality Research Center at UC Davis, of which I'm the director. We have about 70 faculty affiliated. We do a lot of different work in the center. We run the Improved Network and the Chemical Speciation Network for the federal government, which is the primary PM 2.5 chemical composition monitoring network for the whole country. Okay, so we process about 120,000 filters per year through my lab, to the central laboratory. We also work on mathematical modeling of air pollution. We talked about that yesterday at lunch, and we'll talk about it more, I hope. We do a lot of work on physics and chemistry of aerosols, including developing new instruments, of which one reason I'm here is to help take one of the instruments we've developed and translate it here for your use. And then the health effects side, very important, pulmonary, cardiovascular, and neurological health effects. The history there is that when people started getting concerned about air pollution and the health effects of air pollution, of course, the first place you look is the lung, right? I mean, that's where the stuff goes. And yeah, there are some things going on in the lung, and we have to take that seriously, but it kills very, very few people. More research, we find that what's killing people is PM 2.5, and it's a cardiovascular event, myocardial infarction. Okay, so people have heart attacks and they die. It's such a small percentage of the population, though, that it's very hard to know what's the mechanism, what's going on here, why is it? Much less work, although more recently, has gone into the neurological health effects, which may end up being one of the causes for the heart attack, right? Because of the autonomic nervous system, which runs the heart. If you're affecting the brain, you're affecting the autonomic nervous system, and that may affect how the heart works, if it's working at all. Okay, so not clear, that's true, but we're approaching it. And then we have a conference program, which Aron mentioned, the two in Pasadena and Riverside, but many others. And so we have run conferences, roughly 6 conferences or so a year in the US, but also in other countries on air pollution and related topics. Okay, so we're going to focus on traffic-related air pollution. And one reason I do this is because I like to work on what the health effects are of sources of pollution. And the reason why is because that's what we can control, right? That's the only thing we can control. If we don't emit it, we don't have an air pollution problem. So if we can associate an emission, a source of air pollution, with a health effect, then that gives ammunition to EPA, to other regulators to reduce those emissions. So that's the goal of this kind of research. Families that live close to busy roadways have a huge array of health problems at a higher rate than people who live just maybe a few hundred meters away from busy roadways. And you can see the list here. I will focus just on autism and Alzheimer's, but all of these are being investigated, and by folks, we have a proposal. We have a work we're doing right now on cardiovascular health effects, also at the same facility, and we will be submitting proposals on Parkinson's disease in October of this year to NIH. Parkinson's, I've avoided Parkinson's until recently because it is if you're a smoker, you're protected from getting Parkinson's. That is, the rate of Parkinson's among smokers is lower than in the general population. And so I'm like, no, I don't want to go there. But we were recently approached by funding agencies saying, you guys are doing such a great job on autism and Alzheimer's, you got to do Parkinson's too. So when they invite you, you go, okay. So the first question is, how do we do exposures? How do we expose let's just focus on Alzheimer's disease. Alzheimer's disease is something that usually occurs in old age. Sometimes you have a genetic predisposition and you can get early onset Alzheimer's. It's a terrible disaster. It could happen in a 40-year-old person. Okay, but that's very rare. Okay, it's much more common late onset Alzheimer's. And the question is a lifetime of exposure to air pollution from traffic increasing the severity or reducing reducing the time or making it early and earlier onset because of that exposure? Well, we're going to do this work in rats. Now, a rat lifetime is like 2 years. Okay, so we need to expose the animal to air pollution for like a year. No one ever exposes animals in a laboratory setting to air pollution for a year. Okay, that's just not happening. But we have to do that. And so I'll tell you how we do that. The ways we have done experiments exposing animals to air pollution is bolus exposures, where one way is you just take a droplet of you collect the PM, you extract it into a liquid, you put it in the nary of the rat. They reflexively inhale it. It goes into the lungs, and then you look and see what the health effects are. That's a very acute exposure. That doesn't represent anything like what you really are exposed to, except for those occasional buses that come by, and then you get that kind of fast, whatever, acute exposure. We've done longer exposures with concentrated ambient particles. So these are devices which we have built and we've also purchased, where you bring the air in and it concentrates the particles in the air. The problems with that are they're hard to run for a long time. We've done it for weeks, but the technician has to be there keeping it running like all the time because it has a tendency to break or fail somehow. It doesn't concentrate all the particle sizes the same. It prefers the larger particles to the smaller particles. And it doesn't concentrate the gases, which may be part of the problem, because the vehicles in this case are emitting gases and particles. And we don't know which of those phases are the ones that are responsible for the health effects. and then we can do laboratory generated particles. So you can put an engine in your laboratory, you can generate exhaust, and then you can expose, but it's not the same driving cycle. It's not the same realistic situation. So we're going to try to do things as realistically as we can. Okay, so we're going to build a facility right next to a major roadway. We're going to build, it's a vivarium. It's a building that houses rats, or mice next to the roadway. And we're going to take air from that roadway and we're going to expose the animals to it right there, right next to the roadway. So there's no issue with transporting the particles or the gases or whatever. We expose to everything. So it's almost so there's very minimal background here because there's very little else going on. It's a roadway that's in a valley. It's a tunnel coming out of a hill. I'll talk about the tunnel more. There's nobody living there. It's just all by itself. It's a tunnel that goes between Oakland, California and Orinda, California. And I'll talk more about that. It has a realistic fleet, right? So we talk about diesel trucks, we talk about passenger vehicles. We need to talk about a realistic mix of the different kinds of vehicles that are on the roadway. And it really is, the idea is to model the same kind of exposure that people get when they're living in an apartment building or a house near a busy roadway like this roadway. And it's nice because it's continuous, stable, reliable. It has the same dynamic, in the daytime there's more and in the nighttime there's less, and rush hour there's more and otherwise less. And the weekends are different than the weekdays, all those things that are realistic to our lives. And most importantly, it's an unlimited source because the cars just keep going. Now, of course, in California, there are more electric vehicles every day, which is terrible for our experiments. Okay, so here we are. This is called the Caldecott Tunnel. About 60,000 cars per day. There are 4 bores. Okay, so there are 4 tunnels for these cars. 2 of them go to the west and 2 of them go to the east. Each one has 2 lanes. So there are 4 lanes going east and 4 lanes going west. The eastbound lanes are uphill, which means the cars and trucks are working harder. So we want to work there, right? Because going downhill, I take my foot off the gas and I just coast. So there's nothing, you're not emitting much there. Bore 1 is cars and trucks, and bore 2 is cars only. The experiments I'm going to talk about today are from bore 1 only. We have just finished experiments comparing bore 1 to bore 2 to get the difference between the light duty and heavy duty vehicles. We don't really have results yet for that. That's still being analyzed, but I'll talk a little bit about it later. During rush hour, it's not really a freeway or a highway. It's more like a parking lot, which I think you're used to here in Mexico City. Yeah. I had this brilliant idea. So first of all, it took me a year of lawyers to get permission to do this because we're right next to the freeway. There's all kinds of concerns about, causing a traffic accident. Or, one crazy lawyer said, what if rats get out and get into the tunnel? And I'm like, you think there might be some there already anyway? So I had this brilliant idea. So first of all, they said, okay, so after all this year of negotiation and lawyers signing forms, blah, 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 we got permission to do it. And I said, okay, we're gonna have to work. And they agreed to shut the tunnel for us all night for 2 nights in a row. Now, only 1 bore. So at night, the traffic going east, which is this direction, can go in 1 bore. 2 lanes is enough in the middle of the night. In the daytime, forget it. Okay, so they shut 1 bore on one day and another bore on the other day. So we had 1 night, not day, night. We had 1 night to install everything for 1 of the bores and 1 night to install everything for the other bore. Okay, so I had this brilliant idea. I was invited to give a talk in China. So I'll fly to China, my internal clock will reset, and then I'll come back and I'll be ready to work all night. But I got a sinus infection because the air pollution was so bad there. So I worked all night with my clock correct with a sinus infection. And there I am right there. And so we have, all these lights. We rented these big, lights and we illuminated the whole place. They shut the tunnel and we ran these huge ducts, these 8-inch ducts from both of the 2 bores over the 2 nights. And my crew, my research staff that were doing all this, I was up on the top. My son even helped me out. He came one night. But my research staff that were actually down on the tunnel, they were right here working on this area here, but you can fall onto the tunnel, onto the pavement there. And so I took everybody to the climbing gym to get all outfitted, and we roped the whole thing up so that they were all hooked on all the time, and all of the tools were tied to their belts, because we don't want tools falling on the roadway either. Okay, so a little bit of a tour. So there are 3 rooms in the building. The first room has air. The air comes in through these ducts through what used to be a window and is now boarded up, and that's where we bring all the air in. And then we have air pollution samplers and monitors and all kinds of stuff here. We also have to have filtered air. If we're going to expose animals to air pollution, we need to have a comparison. So we have animals here in the facility that are exposed to clean air also. And so we have filters and catalytic converters to get rid of the NOx and VOCs and activated carbon, all kinds of stuff to clean up the air as much as possible before the filtered air animals get exposed. Okay, so here's where the staff were. They were all on this shade. It's a shade screen. So when you come out of the tunnel and your eyes have dilated because of the darkness in the tunnel, then there's this shade screen when you come out that sort of filters the light. And so your eyes adjust before you go out into the daylight. And so we did all of our work on that structure, and that's where everybody was roped on there. And you can see there's the building, and there's the ducts going up the side of the building, and you can see, right, one of them goes here, that's bore 2. One of them you can't really see, it goes to bore 1. And so the traffic is dragging the air in the tunnel in this direction, in the eastern direction, which is this side. And we put the ducts right there above the roadway so that it gets the air as it's been dragged out of the tunnel into that face of the tunnel. Okay. And here they are enjoying their life. So there's little rats in there. We have we have a huge number. We have like 216 cages for the rats. Okay, so we can have 216 adult rats, but if they're younger, we can put more than 1 per chamber, and we can have many, many animals in there to do many different kinds of experiments. So 6 chambers with different kinds of air, 36 cages per chamber, filtered air, tunnel air, and then we can do gases only or particles only, and those experiments just finished. And I won't be able to talk much about them because we don't have the data yet. And again, bore 1 is light and heavy duty, bore 2 is light duty only. And this has all been certified by all of them. We built prototypes of this in my lab before we built it at the facility to get all of the animal care people to sign off to make sure, yes, we're okay with all this. We're going to give you approval. Okay, so let's get some data. So here's PM 2.5. Let's look at that. And you can see, and so we have here's the trap and here's the filtered air. And you can see the filtered air is very low, as we would hope. And the trap is much higher. Okay, right? These values here. So we have PM 2.5 and TSP. So that's good. You can see that it doesn't change that much during the week. The weekends aren't really much lower than the weekdays, which is a little bit of a surprise for me, but it's a pretty busy place. Oh, one important thing here is that of the guidelines. So here's the PM 2.5 concentration, and the trap PM 2.5 is less than the WHO standard. Okay, that's super important. Okay, so that looks like clean air, right? That is, WHO should say if you're breathing 10, then you should be fine, right? Well, we're going to see that that's not the case, okay? The reason why the concentration is so low is because the particles being emitted are tiny, right? And so they don't have any mass. The number concentration is crazy there, but the mass concentration is very low because the particles are so small. Okay, so let's start talking about the health side. That's the exposure facility, and it was the same facility used for the autism studies and for the Alzheimer's studies. So autism is diagnosed by behavior, and it's diagnosed typically not until age 2 or 3 in human, when you see the behavioral differences start to appear. And it's a disaster for families and for the kids. For a long time, it was thought that there was many misconceptions about autism. I mean, the original diagnosis where it's the mother's fault, a terrible disaster for those poor women who were blamed on their kids having autism, a disaster, really, totally wrong. For a long time, it was thought it was genetic. And anybody talking about environmental health effects on autism were just, it's nonsense. But then you start looking at twin studies. They should have the same genetics. They may not have the same epigenetics, if you understand the difference, but they have the same genetics. So it's not complete penetration, right? It should be 100% if it's genetic. It's only 60 or 70% for identical. It's still pretty high for fraternal, but something else is going on here, right? So these suggest that there's environmental factors. Now, it could be diet, it could be air pollution, it could be who knows what, but it's not just genetic. That's the important thing. And this is a real disaster. So Irva Hertz-Picciota, she's at UC Davis. She's in public health at UC Davis, and a colleague of mine, we work together. She published this paper of just in California, the annual incidence. And I mean, this is just insane, okay? So some of this increase is due to different ways of diagnosing autism. Some of it is even crazier than that. I was talking with one autism doctor about this, and he said that, so in California, if you have an ADHD diagnosis, right? Do you know ADHD, right? It's hyperactive, okay? ADHD and autism are closely related. Okay, that's kind of on the spectrum also. If you have an ADHD diagnosis, you get this much help from the state. If you have an autism diagnosis, you get this much help from the state. So parents ask their doctor to give an autism diagnosis for their children so they get more help from the state, which is part of the reason why the diagnosis goes up. Okay? But if you get rid of all those things like that, it's still going up a lot. And this is really scary. And California is not the worst place in the world. South Korea is the worst place in the world. The incidence of autism in South Korea is nuts. Okay. So then there's growing evidence in the literature that autism may be related in other neurodevelopmental disorders, NDD, neurodevelopmental disorder, may be related to TRAP or other air pollution sources. So here's carbon monoxide, right? The South Korea, I just mentioned this, incredibly terrible high incidence of autism in South Korea related to NO2 and PM. NO2, though, it may be the cause, but it's also a tracer for diesel trucks, which emit a lot of NO2. Okay, so you don't know if it's because of the diesel exhaust, other things that they didn't measure, if it's NO2 is the actual cause or whether it's just correlated to the cause. Anyway, lots of growing evidence in literature, which then motivates the studies that we're doing. So the trouble is, if you live near a busy roadway, maybe it's because the real estate is cheaper there because no one wants to live there, which maybe means that you're poor, which maybe means that your diet is poor, or you have more stress in your family, or many other things, right? Rats have the same socioeconomic status. About the same as politicians in the US, I would say. And so we can do experiments like this, which eliminate the diet. The diet is the same, socioeconomic status is the same, stress, all these things are the same for everybody. The only thing that's different is filtered air or TRAP exposure. Okay. So the endpoints we're going to look at, and I'm not going to do everything. All this is published. So if you want more information, I can show you the publications. But we're going to look at behavioral outcomes, and that's Jill Silverman. That was what she did. And we'll look at neuropathology outcomes. That's what Pam Line and her lab did. So with 2 different groups working together. And you can see they're playing all the time here. Whoops, okay, here we are. They're playing, they're wrestling, they're having fun. But you can see differences in how they do it if you understand rat behavior, which is what Jill's expertise is. Okay, so the rats were impregnated and they were brought out to the tunnel 14-day GD. That's gestational day. That is 14 days after insemination, okay? And then at postnatal day 0, that is when they're born. So they're exposed in utero for 2 weeks, right? And then they're exposed for 50 days. And then we did some tests at 21 days and some tests at 50 days. And I'll show you again some of the results. So now we had 3 different cohorts. We had one that was at the tunnel, exposed to tunnel air, one at the tunnel exposed to filtered air, and one on campus. And I pushed so hard to get that campus one. And Jill and Pam were like, "You're wasting our money, blah, 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 blah." I'm like, no. Because the other thing that can affect behavior is noise and vibration. And if you are next to that tunnel, when these big trucks come by, there's a lot of noise and a lot of vibration. And I wanted to have one in a quiet setting so we could differentiate how much of the health effect, the neurological health effects, were due to the air pollution and how much due to the noise and vibration. And they finally said yes, and boy, it was good we did, and I'll show you why. So first of all, just to look at some, and I'm sorry for this, the size of the font here, but this basically is saying that the rats that were exposed didn't grow as well, okay? So these are the males, these are the females, and autism is much more prevalent in males than in female, so something to keep in mind. And it will show up, we'll actually show that more later. And you can, so there's all kinds of different data here, body length, body weight, all kinds of stuff here with growth and behavior. So here's the growth metrics, and you can see they're lagging here. And here is the behavior, so rooting behavior, forelimb grasping. So these, babies do the same thing. If you put your finger in their palm, they hold on. That's a reflex. Same thing with rats. They want to hold on, and they want to find the teat to drink, right, to nourish themselves. And so they're not looking around. The animals that are exposed aren't looking around as much as the animals that had filtered air. They're not trying to find their nourishment. Okay, that's a bad sign. So the kids are not hitting their milestones. Okay, so here's where the cohort at UC Davis that was not getting the noise and vibration comes out. So just like with human babies, if you separate them from their mother for a while, they don't like that, right? They like the warmth, they like the nutrition. All those things they're hardwired in human and in rat. And they cry, and so do baby rats. Now you can't hear the baby rats cry because it's beyond our range. So we can hear up to about 20,000 hertz. They cry at about 40,000 hertz. So you just can't hear it. But of course, we put a microphone there, and we can record that, and we can see that they're crying, or not, if they were exposed. Okay. So again, male here, female here, significant difference in the males, but the comparison is to the lab that the significant difference is, because they didn't get the air pollution or the noise and vibration. Okay, so there's some kind of combined effect of the air pollution and the noise and vibration that is making it worse. Okay. And this is almost significant. I mean, the official way to say is 0.05, right? We got 0.055, so it's not quite significant in females, but this is actually expected because in the human population also, women don't, or girls don't get an autism diagnosis. Like 1/4 as many girls have autism diagnosis than boys. Okay, so we see some behavioral changes. We see some cell proliferation changes. I didn't talk about that because it's a lot of detail looking at slides of cells and stuff. And we see sex differences just like we do in the human population. So that's one of our results here related to this autism and neurodevelopment disruption. Okay, so now let's move on to Alzheimer's. So chronic neurodegenerative disease worsens over time. A lot of dementia cases are Alzheimer's, but some of them are not. More prevalent in women than in men, much more. And I have some theories about that too, which we're exploring in a human study that we're doing right now. Again, increased incidence in families living near busy roadways, which is why we're doing the study. And it takes years to develop, which is why we have to do things at this tunnel. The animals have to live there for a year before we're going to see results. Let's just get all these out here. So very few are solely genetic. Very few cases are just solely genetic, right? And it's sort of the same thing with, in a mirror in a way with autism, right? Is this genetic? Is it environmental? Very little of it in Alzheimer's is solely genetic. There are some, and we'll talk about this a little bit more, there are some mutations which predispose for sure. So a lot of environmental factors involved. We don't know all the different environmental factors, but of course, because we see families living close to busy roadways having a higher incidence, we think, okay, maybe there's something going on. Could be the noise and vibration, could be the diet, all those things I just talked about that are maybe cofactors to living near a busy roadway. But the bottom line is, with rats, we can try to tease this out. Okay. So again, we have a shortcoming in data in animals because there are no exposure facilities in the world like this one that can do this. Okay, there's one being developed with our assistants in Hong Kong, at Polytechnic University of Hong Kong, and they are conveniently located right next to the entrance to the tunnel in Kowloon to Hong Kong Island, and 60,000 cars per day is nothing. They're doing 2 and 3 times that. And so I've been there helping them and getting their setup going. So we need to understand all of these things, right? Chronic exposures, what's the effect of sex difference? Why is there sex differences for this? Is an exposure difference? Is it some inherent in that chromosome? Which pollutants are responsible for this? We just finished studies where we did bore 1, which is a light duty vehicle, versus bore 2, which is mix, and where we did gases and particles separated, but we don't have the results yet. And what's the mechanism? Basically, what's going on here? We are currently doing a study where we are using a drug that is being proposed to the FDA, which is supposed to counteract Alzheimer's, and some of the rats have that in their feed, and so they're being exposed to that every day. They're getting that drug, and we're going to see whether that counteracts the effect of the air pollution. Okay, I think I talked about all this. Okay, so what we're going to do is we're going to look at something called the gene-environment interaction. So we're going to look at, we have rats that have the human early onset Alzheimer's genes in them, in their genes. Okay, there's these 2 here, and so these rats will get Alzheimer's, and we'll be able to see that in their brains and their behavior. So what we're going to look at is severity and whether the time course changes due to the exposure. Sorry. And so we're going to use this, we talked about this. It's already been shown that this Fisher, this is a Fisher 344 rat that then has been genetically modified to have these genes in it. It's already been shown that the Fisher 344, when exposed to air pollution, develops brain lesions and other indicators of Alzheimer's. Okay, so that's our background, our genetic background. Okay, so we breed animals that are both regular rats, but Fisher 344s, and ones that have these Alzheimer's genes in them. And we breed them up. And after a month, we put them out at the tunnel. And then they're exposed from then on. And we take samples at these different time points for histology, and then also for behavior at 10 and 15 weeks. We're looking at plaques. Plaques are seen in the brains of Alzheimer's patients, but not 100% associated with Alzheimer's. But a lot of people talk about plaques, and so we have to do that too. Microglia in the brain, very important. We'll talk about that a little bit. Astrocytes and neuronal cell loss. We'll look at various things. I'm not going to talk about everything here, obviously. Not enough time. Yeah. Okay, so let's go back up here. So we have the tunnel, we have filtered air. So we have the trap exposed animals, filtered air. We have both wild type, that is no irregular genetic background, and we have the ones with the Alzheimer's. And then we have sex. We have 8 different groups. And then we have to have about 15 rats per group to do the full study. Okay. Now, one of the things we did not expect to see, many things we did not expect to see, is particles in the brain. Now, and especially in this location. A lot of the literature, mostly from occupational exposures to nanoparticles, showed particles in the brain in the olfactory bulb. How am I doing on time? 20 minutes, okay. So I'll make it. And that it made sense because you breathe in these particles. Your olfactory bulb is basically an extension of the brain into the nose. There's tons of neurons there for your sense of smell. Now, our sense of smell is terrible compared to a rat or a dog or whatever, right? But still, and you can see that if you ever look at the structure of the nose, we have very few turbinates in our nose. They have tons of turbinates in their nose, making much more surface area for this transport of particles and gases to the neurons. We didn't see any particles there, which is completely counter to a lot of the literature. And we were very surprised about this. We did see a lot of particles in the hippocampus. Now that's very interesting for 2 reasons. One is that hippocampus is central to memory, which of course, Alzheimer's is a memory thing, right? The second thing is that the blood-brain barrier in the hippocampus is weaker compared to the blood-brain barrier in other parts of the brain. Okay, so from that point of view, it's expected. Okay, so here we are. Filtered air, tunnel air, wild type, genetically modified. You don't see any particles here. That's, hopefully that's true, right? Because otherwise, what are they doing there? But you do see them even in the regular. You see it, right? You can see these white dots here, right? Those are all particles that made their way through the blood-brain barrier into hippocampus. You see much more in the genetically modified rat. And one characteristic of Alzheimer's disease is a breakdown in the blood-brain barrier, right? Breakdown in the blood-brain barrier, a lot of B's. Okay, and there's the statistics. Okay, those results were published. These results have not been published. They're hot off the press. And this is the work that we did with Barbara Maher, who's from the University of Lancaster. We see, so look at the particles. I mean, this is nuts. These are particles in the brain and hippocampus, okay? But look at that. What the hell? Is that a neuron? Is that a blood vessel? What is that? We don't know. We haven't been able, we have to do a study, which we don't even know how to do, to label the tissue and the particles at the same time with the same imaging technique. We don't know how to do that yet, but we're trying to figure it out. So this is a 1 micron scale, okay? And so, the cells maybe these are axons, I don't know, but something. I mean, look at that. Look at this one over here. There's one right there. Now, this is an 8 nanometer slice, okay? So we just happen to hit I mean, and we have multiple slides like this. It's not just the only one, but you know, the neurons are going every which way. And if you take just an 8 nanometer slice, you can see it, you can image it like this. The neuron has to be in that slice the whole time, right? In order for you to see that. It's crazy. You know, one thought was, oh, it's the blade when it cuts the tissue, dragging the particles along this direction. But then we see this, and we see many other slides with crossing and all kinds of different stuff going. So it's not an artifact of the blade cutting. Okay, now look at this one, 200 nanometers. Okay, so this is now all subcellular, right? This is all within a cell, and you see these structures like these loops and round stuff like this, round, 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 right? Look at that, round. Is that around the nucleus? Is that around a mitochondria? Is it around some kind of organelle within? We have no idea. Again, we don't know how to image this stuff to figure out what's going on. But again and again, we see these structures, and it's like that's down the road. That's coming up. Okay, and then we can do chemical analysis of those particles one by one. Those are like, we're talking a few nanometer size. There's like 5 nanometer particles, and we can do chemical analysis on them one by one. A lot of this is background, but the mercury is not. Almost every particle that we see has mercury in it. What does mercury have to do with vehicle emissions? I have no idea. So we are completely stumped by this, okay? We don't know if the mercury is coming in as a gas and then somehow being attached to the particles, or whether it's already attached to these small particles coming from the vehicle, or whether it's there's all kinds of possibilities. We're totally stumped. So again, these results, we're still working on this. I just wanted to give you a little taste for some of the things we're working on. We also see zirconium, things that are in the catalytic converters, iron, copper, various things you would expect. The calcium is probably just in the tissue. So anyway, okay. Is there a question? No? Okay. So here we're looking at more statistics of this. So again, we're just the cerebellum, not much. There's some particles in here. It's not like hippocampus. And you do see that, as you'd expect, that as time goes on, the number of particles increases because the exposure's for longer and the brain is not flushing them out. They're just staying there. Okay, now for plaques, again, one of these things that people want to look at if they're Alzheimer's people. 3, 6, 10, 15 months, filtered air, trap air, the red are the plaques. So you do see some, as you'd expect, because these animals are genetically predisposed to get Alzheimer's, and so you'd expect some plaques. But look at it's just completely different, right? Completely different. And then the statistics on that. Here's the hippocampus. Let's focus on that, right? So here's the wild type. So this has to do with cell loss. So one of the things that's happening here that we've noticed is that the microglia. So the microglia are cells in the brain that clean up the dirt. Okay, whether there is some infection, or whether they are whether they're pruning neurons that are no longer useful. Okay, these particles make those microglia go crazy. Okay, these exposures, and so they're overactive. And so what happens is you get more cell loss because these microglia are trimming neurons that they shouldn't be trimming. Okay. And so here's the microglia slides. So you're getting these really angry microglia, and even for the wild type rat, you're getting them, but of course not as much as when you have the genetic predisposition. Okay, and then there's the cognitive deficits. So learning, learning and memory deficits here. I'm not going to go over all this, very complicated, lots of data, but the bottom line is the animals also behaviorally have deficits due to this exposure. Okay, so here's a summary slide. And it's not clear. Things are always complicated in biology, right? So here we see plaque deposition higher in 3 months and 15 months, but not in 6 and 10 months. Don't know why. So there's a lot of bad things going on here, but there's also some crazy questions that are brought up that we don't know the answer to yet. So this is this ratio of 42 of this this A beta protein. This ratio is very important for diagnosing Alzheimer's, for instance. Okay, Alzheimer's conclusions. So chronic exposure to trap, right, promotes AD phenotypes. So you're getting both the brain, the histology, the pathology is there, the behavior is there. It's complicated, but so is in the human population too. And microglia activation is probably one of the roots that's causing this problem, right? So they're phagocytes. That is that they're, in the lungs, say, or in other parts of the body, they would be taking a bacteria and basically killing it by oxidizing it, okay? And so microglia are there, to repair infection in the brain, but also to get rid of these neurons that are no longer needed in order to keep things under control. But the particles or something is being transported into the brain that make these guys go crazy. And that's probably one of the roots for the effects that we see. So, and because the particle concentrations that we're exposing these animals to are below the standards that we currently have, we're not protecting human health. And the Environmental Protection Agency, its primary mission is to protect human health. And so the standards aren't doing that. Now in Europe, there's a number standard. We don't have a number standard in the US yet. So overall conclusions, the tunnel facility is incredible for these long-term exposures. It wasn't my idea. It was Keith's idea, one of the people I acknowledged, okay? And it turns out he lives about 10 minutes from the tunnel. Otherwise, it's like a 2-hour drive from Davis during rush hour, which is the time you usually go there. So very painful. But we have to go there all the time or he has to go there all the time to take care of the animals, give them food and water and change their bedding and all this sort of stuff. So all kinds of experiments are possible here. We are currently doing experiments. We just finished experiments looking at the gases versus particles and the light duty vehicle versus heavy duty vehicle, and those are being analyzed. Preliminary results are that the heavy duty is much more toxic, neurotoxic than the light duty, and that whether you do particles only or particles and gases, you get the same results. Okay, so it's likely just the particles then. We are doing experiments now. It took me 8 years to get the funding for this, to look at heart attack, to look at the cardiac endpoints, since that's what's killing people. I don't know why it took me 8 years to get the funding. We tried so many times, but anyway, we have funding, and we are looking at what's called heart rate variability. How much time do I have? 10 minutes, great. We're looking at heart rate variability, which is that your heart doesn't beat at the same rate, it turns out. It just, it moves a little bit faster, a little bit slower, it moves around, When you're under stress, the heart rate variability decreases. And when there's very little variability, you're gonna have a heart attack, okay? So we look at heart rate variability because it's a continuous measure of normal state to highly stressed, close to heart attack state, okay? And so, now the Chao Chen is the professor I'm working with on that. She's the cardiophysiologist, and she's doing that work. And she's done this before, but not at the tunnel facility and using other exposure methods. And she found some results, okay? This experiment is different in that the other person we're collaborating with is Jane Clougherty. She's a professor at Drexel University. And when she did her PhD at Harvard, she discovered epidemiologically that the heart attacks are more prevalent in populations that have high stress and air pollution. It's a combination, okay? So what we're doing is we are stressing the animals and exposing them to the air pollution, okay, at the same time. And we haven't analyzed the data yet because we're talking terabytes of heartbeat data we have to wade through. But just for kicks, Chow sent some of the animals to one of her colleagues that uses echo, echocardiogram that looks at the flow ejected from the heart, way down in the animals that had stress and air pollution exposure, right? So that wasn't even in our proposal. That was just for kicks. Shocking. Yeah, so the tunnel is an amazing facility for learning all kinds of things about air pollution exposures, especially from traffic. They're not substantially higher than ambient concentrations on a mass basis, on a number basis, completely different. And then we have all these results. We have modest but very concerning neurodevelopment disruption or autism results and Alzheimer's results. And studies are underway to answer these questions, which I already indicated. Yeah. Thank you very much.
Tenemos tiempo disponible para preguntas. Si desean hacer las preguntas en inglés para que directamente se las conteste Anthony, bien. Si las quieren hacer en español, tendrán que esperar a que se las traduzcamos.
Or you can just translate.
Or me dice, es que we have a translator.
Okay, should I put this on?
So let's see. Okay. Por favor, vamos a hacer las preguntas con el micrófono porque estamos transmitiendo esta presentación. Entonces las personas que están fuera de este auditorio tienen que escuchar también la pregunta. Adelante. Sí.
Okay. Thank you. That was really interesting. I wanted to know if the composition of your particles a little bit more, because there were a lot of metals, but there were small concentrations, but you also had calcium and potassium. I mean, what do you think is the main composition of these particles? And
right.
Yeah, I know you mentioned that you didn't know where the mercury came from, but
right. Good question. So first of all, I didn't show this. I think I may have a subsequent slide. We did chemical analysis on all the PM we collected, but those tiny little particles that are getting into the brain are a small subset of all the particles that the animals are being exposed to. And so it's kind of a complicated problem because, so you inhale the particles, they deposit on the mucus, and that's mostly water. And so all of the, sulfates and nitrates and all that stuff, it dissolves and it's not going anywhere, right? And then it gets transported into the bloodstream, and anything in there that is lipid soluble, right? That's the organics, that's now going into all the fatty tissue in your body. The only thing that's left is the refractory material, right? The metal oxides, this kind of stuff, or soot, black carbon. That stuff then goes, what you see here is that stuff is circulating in your bloodstream and then finally getting through the blood-brain barrier into the hippocampus. But these particles are so small, and when you're using X-rays to get these, that's all. What I showed you is the kind of stuff we have. We have more spectra like this, but we're talking about, mercury is incredibly common among all of the particles we see, but then we see copper, we see iron, and some of these things you're going to see anyway. Iron in the body, okay, come on, it's all over the place. So we don't know yet. We're getting there. Yeah, but this is a big question for me, because one of my bottom line questions here is, are the catalytic converters responsible for this? Right. We saw zirconium, right? So I've been looking hard for platinum, you know, rhodium, because they're nanoparticles in the catalytic converters, and maybe some of them are getting out. And maybe that's what's, maybe ironically, we solve one air pollution problem and we cause another one. Right. Exactly. She said electric cars. Graciela, hi.
Hi, thanks. Great talk. I was thinking, can you do an estimate of the enrichment factor for some of those metals?
So what you mean by enrichment factor, what do you mean by that?
Well, you have the clean air population, right?
Aha.
Can you do, even though it's not the same brain, they are fed the same brain?
Yes.
Can you find some particles? Because I thought that you saw some particles in the normal population.
We do. That's a good point.
If you do that, and I remember many years ago, people were talking about the transition metals.
Yes.
Is this one of them?
Look, there's two different things here. So the first thing is that when we take thick slices and we look at them with microscopes that can see them, we see particles, and I showed pictures of that, of both the wild type, in the wild type also, okay? But not in filtered air. Okay, so we don't know, we see nothing in filtered air for particles. Okay, so so I don't know how to approach that. We do see, of course, more particles in the genetically predisposed animals because their blood-brain barrier is weaker. I don't know how to talk about enrichment factors, though. It's a good question. I'll have to think about that. The other question is transition metals. So the transition metals are like, there's a few different transition metals out there that have two stable oxidation states, and they cycle between them, typically in a Fenton reaction with ascorbic acid or some other antioxidant. And basically what they're producing is ROS, reactive oxygen species. So OH, whatever. And which is how the microglia kill the bad cells, okay, is they oxidize them. And when you have inflammation, that's what's happening, is that you have a cut, right? And there's some bacteria that got in the cut, and you're getting inflammation there, which is your body's natural response to it. And if it was, and the problem here and in other health effects from air pollution is that there's a couple of things. One is that the macrophages try to kill the particles. They're not killable. So they're just working away, trying to oxidize these particles because they think it's a bacteria or something, or a virus, and they're killing everything around it. Okay, this is true in the lung, this is true in the brain. Yeah. So so now coming back to your question is, the transition metals also produce that. Okay, also produce the same highly oxidative species, OH radical, for instance, that the macrophages produce. Okay, because it's just called a Fenton reaction. Look up Fenton reactions. It basically is a cycle between the two oxidative states, and then one of the states being returned to the other state by reacting with the antioxidant that's naturally in the body. I hope that was comprehensible. Yeah.
Hola, buenos días. Muchas gracias por su presentación.
Wait. Yeah, okay.
La pregunta va orientada hacia que usted en el principio mencionaba el incremento de autos eléctricos en California. Entonces, quisiera saber si dentro de su estudio está consideradas las emisiones de estos autos eléctricos dentro del túnel o es únicamente para emisiones de combustión interna. Gracias.
Yeah, okay, good. Very good question. So in California, where there are many electric vehicles, I think in California, because of various incentives, there are more EVs in the car population than in other parts of the US. Still, the percentage is very small. There are no tailpipe emissions because there's no tailpipe, but the electric vehicles weigh a lot more. Okay, so let me back up. There are four categories of emissions from vehicles. okay? One of them is the tailpipe. One of them is tire wear. Another one is roadway wear. And the third one is brake wear, okay? All of them can be problems. For electric vehicles, you remove the tailpipe emission and you remove the brake emission, because even though they have brakes, they're only used during emergency stops. For regular slowing down, they regenerate the energy, the kinetic energy of the vehicle into the battery. Same thing with hybrid vehicles. They don't use, the brakes last forever on EVs and on hybrids, okay? You still have the tire wear and the brake wear. I'm sorry, the tire wear and the roadway wear, okay? Those particles are huge. And it's an abrasive process, not a combustion process. So the particles that we're seeing, these very, very small, like few nanometer particles, I don't think you're gonna get much emission from roadway wear and tire wear in those particle sizes. That doesn't mean there's not a health problem, because the tires have transition metals in them, like copper, and okay, and so do the brakes. And now there are more regulations in the US regulating what kind of metals you're allowed to have in tires because of this. The electric vehicles weigh more, a lot more. And so there's more tire wear and more roadway wear. So we don't know what the health effects and problems of the EVs are. But because they weigh more, they do emit more roadway wear and more tire
wear. Hi, Anthony. Thank you very much for this wonderful presentation. I'm just curious about nanoplastics. I know they might not be traffic-related particles, but they have been reported in different parts of our body, and I'm not sure they could make it to the brain. So on these beautiful samples that you have collected for the last, on this project, did you get any sign or nothing about that?
We did not, but it's not because, it's because the analytical techniques that we're using wouldn't see them, because they're hydrocarbons, right? And the whole body's hydrocarbons. And so you're not going to be able to distinguish what hydrocarbon is there from the plastics and what hydrocarbon is there that's supposed to be there, because it's how your body is built. Yeah, so we can't see that with the current techniques that we're using. But I know there's a lot of, I just got to make a comment here about nanoplastics. There's a lot of interest in the nanoplastics and health effects. And when I talk to people who work on this, I ask them, what's the difference between nanoplastics and fiber, nutritional fiber? They're both not digestible, right? So why do we care about one and not the other? And they have not been able to give me a good answer for that yet. And I'm not saying, I mean, the nanoplastics may be terrible. I mean, I don't know, but that's a fundamental question, right? It's like the doctors say, you should eat lots of nutritional fiber, right? Okay. Should I eat nanoplastics instead? I don't know.
Hello, doctor. Thank you. Well, I have so many questions, but I want to focus in two main issues. One is the stress, and have you ever measured cortisol or some hormones?
Cortisol, cortisone, yeah. Sure.
Because, I mean, this is obvious in the lab group and the group.
Yeah.
And the other thing, have you measured cytokine in the brain, in those brain areas?
Okay.
I mean,
yes.
to see what's the profile,
right.
Inflammatory, pro-inflammatory, and also if there's some mechanism of immunosuppression in both cases.
Right.
And regarding to plastics, I just want to say that the problem maybe, well, the particle is a problem because they accumulate just like the PMs, but the plasticizers are...
Yes, phthalates.
Yes, because they release and they are endocrine disruptors, and they can be immunomodulator and...
Right, phthalates are everywhere. And that's because they're plasticizers. And I get that. But by the time the plastic particles have gotten to nano size, I think all the phthalates have evaporated.
No. That's the main problem.
I don't know.
Thank you.
But I mean, I agree. The phthalates are terrible, and plastics have them in them, and that is a possible reason why we should be concerned.
Yeah, because plastic sensors are associated to depression, to anxiety.
No question. Yeah. Okay, so now there were 3 questions. I answered the last one. First one was about, what was the first one? Cortisol. Okay, cortisone, whatever. So when we submitted this proposal on the stress, the stress and air pollution causing heart attack, of course we put that in there, but we didn't believe we're going to see anything, because if you really look at the literature, there's no correlation. People talk about this all the time. Cortisol, cortisone, whatever, stress hormone. We don't buy it. Okay. Controversial. We put in the proposal because some reviewers are going to look at that and they're going to hurt our proposals, because the common thinking is these are stress, right? These are stress indicators, but no. If you look carefully at the literature, it's no. And so instead of just rebutting that in our proposal, we say, okay, we're going to measure it. There was a second question. Now forget, because you asked so many questions. What was the second question?
Cytokine profile.
Yeah, and beyond me. Yeah, I might have a slide. Let me see if I got a slide. Yeah, I'm sure we did it. No, I don't have it. Yeah, so it's so much data. No, okay, no, sorry. It may be in the papers. So if you email me, then I'll ask my neuro colleagues.
Sure, thank you.
Just to remind you that Karen is part of hopefully of the next project you're going to be
involved in. Okay,
so
great.
You're going to be able to discuss a lot of things, I see.
Okay, great.
Yo? Okay, thank you. You call us to notice about mercury.
Yes.
But you say nothing about lead.
Yes.
That is abundant, or is just marginal?
It was in the spectrum. There was lead.
So,
but we don't see it very often. The mercury we see in almost every particle, but the lead is very infrequent. So lead has been taken out of gasoline in the U.S. a long time ago. There's plenty of lead on the street still, because that's where it ends up, and so it gets resuspended. But most of those particles are very large, and so they're not these nanometer-sized particles. So occasionally there is some lead, but not very often. Yeah. But you guys have seen the movie or read the book Alice Through the Looking Glass or Alice in Wonderland, right? And you remember the Mad Hatter? The Mad Hatter, he made hats using mercury to form the felt into the hat, okay? And then they all went crazy. So maybe mercury in the brain isn't a good idea.
Okay. Thank you for such an interesting talk. It's been quite revealing.
So raise your hand so I know where you're coming from. Over there. Okay.
Yeah, that's me. You mentioned something. There is growing concern about historically we have focused on linking the effects and particular matter, but considering mass.
Yes.
We have not focused, or we have just recently pay attention to link the effects between number of particles
exactly
and the health effects. Mm-hmm. And that's, well, for example, here in Mexico and everywhere, being observed that with the development of the new technologies, the mass and the emissions have decreased, but the number of particles increased even exponentially.
Yes.
And that's a recognition, of course.
Yes.
So in your opinion, and in your experience, in this case, in this kind of in your studies, have you considered not mass but number of particles, and how would be the perspective in the short term if this kind of regulation should be implemented instead, or maybe having both mass regulations and number of particle regulations?
Yeah, good question. how do I answer this? So number is difficult because generally speaking, the number distribution increases as the particle size goes down, right? The larger the particle, larger particles, a few number, small particles, lots of number, right? So the question is, where do you stop? If you say, okay, we're going to look at particles that are larger than, say, 10 nanometers, you get one answer. If you say 5 nanometers, you get a different answer, right? So this is a fundamental problem. The Europeans have worked on this a lot. And they have a number regulation for vehicles. So in Europe, the number vehicles are regulated or restricted by the number of particles they emit, okay, in order to help with this problem. But there was a lot of fighting about where that line is, both from a point of view of how do you measure it when they get really small, and from a point of view of health impacts, which we don't even know. I mean, we're getting evidence for this study, but there are very few studies. I mean, this is the only tunnel like this in the world that's got this going on. This is the only study. And so you can't do regulations based on one group study, right? You have to have many people getting the same answers or similar answers. And hopefully people in Hong Kong will get good results from their work. And then other people hopefully will do the same thing. So what drives regulation is epidemiology. But epidemiology is an association. It's a statistical association. When PM 2.5 goes up, we see heart attacks go up. When PM 2.5 goes down, heart attacks go down. Okay, now we have a PM 2.5 regulation. But if we weren't measuring PM 2.5, we wouldn't see the result. So it becomes a chicken and egg problem, right? If you don't have any epidemiology saying there's a problem, then how do you justify spending money to measure? But now with this, hopefully there'll be funding to say, okay, let's start measuring number in many cities around the US or other places around the world and seeing if there's a correlation to, say, heart attack or Alzheimer's. The other problem with number is PM 2.5 doesn't vary that much, even by a roadway, because there's not that much, because the particles are so small, right? You don't see PM 2.5 going up by a roadway, or very little, okay? It's just kind of uniformish. Number concentration is not like that. Number concentration is very dynamic, very spatially heterogeneous. So it makes it much harder to do an epidemiological study. When you're 300 meters away from the freeway, you're not going to see the number concentration like if you're 100 meters, which means you may need to have tons and tons of number instruments in order to get any kind of statistics. It's hard. Yeah, full-time work for us, all of us. Good job security.
Oh, what great your talk. Thank you. I try to understand the role of your transgenic animals in your experiment. They have some special genes, or which is the?
They have 2 of the genes that are in the human population that predispose to early onset Alzheimer's. So they're the same genes that we have, I mean, that hopefully only a very few of us have.
And the particles are able to arrive to the brain?
Yeah.
Oh, it's terrible.
Yes, it is terrible.
Thank you.
We are doing a study now in collaboration with the Alzheimer's Research Center at UC Davis, where they have collected the brains from people with Alzheimer's and without Alzheimer's through all over Northern California. And we're going to start looking for particles in the brains of the Alzheimer's patients. They're deceased. They're all deceased. I mean, there's some that are still alive in the study and some that are deceased, but we have their brains. They have their brains. And so we're collecting slices now and looking for particles in human to see whether we see the same thing as we see in the rat.
For the animals, it is a chronic effect for all them?
What do you mean by chronic effect?
The effects.
They have Alzheimer's pathology and symptoms, right? And that lasts. Yeah.
Thank you.
Sure.
Interrumpo. Tenemos una pregunta del internet. Es de Jaime Contreras. Could the effects be different in various kinds of rats? American, African, black, white, brown rats? That's, I think, the gist of the question is if besides the sex, also there are certain people that are more predisposed.
Yes.
Certain groups of people.
Yeah, we have not done those experiments, but sure, it could be. Yeah, it could be that the Fisher, the reason why the Alzheimer's genes were put on top of the Fisher, it's various, there's brown Norways, all kinds of different rats out there for laboratory use. The reason why they were put on top of the Fishers is because there already was information in the literature about the Fishers already having a response. But it could be that other ones have a worse response or no response, or I have no idea. Yeah.
And just one comment in English, okay? There have been some experiments in Mexico City also with particles in the brain.
Yeah.
The center, our institute, participated in one study. Ricardo Torres Jardón participated in a study with dogs in Mexico City that they found they have an excess of magnetite in their brain. All right. So it's also part of the cumulative study of this type of problems in urban settings.
Yeah, thank you for saying that. I should have said that.
Oh yes,
yes, because Mexico City, it's cleaner now than it used to be, but it used to be really terrible, like Los Angeles.
Yes.
And there have been studies here of Alzheimer's, and especially the brains in dogs, which are easier to acquire than brains in people. They see particles and they see pathology, Alzheimer's-like pathology. Yeah.
Okay. So, thank you very much for coming. You are, I think we're going to see you very often here.
Good.
You and your wife. And he already went to a lot of museums, but Mexico has so many that I think you have another 3 or 4 years of using the wonderful museums we have in Mexico City. Yes. So please have a round of applause for.
Thank you.
For Anthony. I'm going to present the diploma for you. Thanking you.
Thank you.
And also.
You want to see this for the photographers?
Yes, we're going to take a photograph. And also one.
Toys. Nice. Great. With UC Davis colors.
Okay. So please,
gracias.
Sound of a car.